Welcome to a very warm afternoon. Dear audience, dear colleagues, dear students, Bagdifizenz, dear Bernhard, dear Dr. Esvelt, dear Kevin, may I say so. As one of the two directors, the rector already alluded to this, of the Marsilius colleague, I cordially welcome you all to this lecture, which runs and was advertised with a short title, Wisely Engineering Shared Ecosystems, but the more detailed title gives a little bit more of the information, leveraging gene drive. And this is the topic, gene drive to ensure open and community-responsive genetic engineering research. Together with Bernd Schneidmüller, the rector already briefly alluded to this, we have recently launched a new format of interdisciplinary debate with the aim to address controversial societal issues from different perspectives, running under the name Marsilius Controverse. So in early May this year, we had a lively exchange between a molecular biologist, a theologist, and a public health colleague on the topic, this was already mentioned, maybe design human being. A topic which addressed the new options for modifying genomes provided by CRISPR-Cas technology-based genome editing. In that very lively debate, the focus was on medical applications and related ethical issues. Today, Kevin Esvelt will introduce us to another perspective, again opened by CRISPR-Cas technology, the gene drive. The relevance of CRISPR-Cas has been compared to that of nuclear fission. As we are going to learn today, gene drive is about to deliberate alteration of entire populations of wild organisms in natural ecosystems. And as some of us already know, and as we all can imagine, these new perspectives have already initiated a passionate public debate on risks and chances of this new technology. Here, Kevin Esvelt kicks in. He's not only a knowledgeable scientist, but also a staunch advocate for opening the so often closed shop of science. Behind his engaged communication initiative is a conviction that the most important ecosystem to be engineered is the scientific enterprise itself. Consequently, in his opinion, the most important application of gene drive is not, this is very important, not to eradicate malaria, this is very important, not to eradicate other diseases, no, it's to change the way we do science. So, shaping the world in a responsible way. In free democratic societies, this obviously requires enormous communication skills when entering an educated public debate on complex scientific issues. But it also may come into conflict with massive corporate interests. In this respect, it is of interest, at least it was of interest to me, and here I quote an article from Michael Spector in the magazine New Yorker, saying, Kevin Esvelt, and we are going to see whether it's still like this, wears his political opposition to corporate science like a neon badge. So we are very curious if this is still the case. But now a few words on his very, very unorthodox career. After graduating from Harvey-Mudd College in engineering school with a strong humanities program, Kevin Esvelt moved to Harvard to the laboratory of David Liu, a professor of chemistry and chemical biology known for his work on directed evolution. While graduate students normally attempt to publish as soon and as often as possible early papers, Kevin Esvelt did not publish any paper in the first 5 years. I hope it's correct, I cite correctly. As Professor Liu recalls, and here I quote again from this article, Kevin told me the day we met that he wanted to forego smaller projects to accomplish something of genuine impact. Kevin struck me as somebody who had all the skills and all the ambition he needed, but he had also had just the right amount of naive fearlessness. You need some courage to wait for a number of years. In the end, his PhD work earned him an award, one of the country's most prestigious, in fact, prizes for graduate research in the biological sciences. Then, from 2011 to 2015, he worked at the Harvard Medical School as a WIS technology development fellow. During this period, he developed several key technologies that utilize the RNA-guided CRISPR-Cas nuclease for genome engineering and regulation. The most notable of these is the RNA-guided CRISPR gene drive that he also will talk about today, which may allow to deliberately alter the traits of wild populations. In 2016, so very recently, he joined the MIT Media Lab as a professor of media arts and sciences. At the MIT Media Lab, he leads the Sculpting Evolution Group, which explores evolutionary and ecological engineering and responsive science. So it is perhaps fair to say that Kevin Esvelt has a strong touch for interdisciplinarity, and he certainly is both a scientist and a public educator. When trying to describe his attitude towards nature, I take again the liberty to quote from the New Yorker. "Natural selection is terribly immoral," Kevin says. Unlike Rousseau, he sees nothing blessed about man in his natural state. In fact, romantic notions of a natural world defined by innocence and harmony repel him. The idea that nature is the essence of goodness, is purity and truth, is so foreign to my perception, he says, it's even difficult for me to conceive how people can think like this. There is such a fantastic degree of suffering out there in the world. This is definitely a drastic statement, but perhaps a statement that is worthwhile to reflect on. As we're all aware of, and the director already alluded to this, in German, gene technology, in particular for transgenic crop plants, and the plant science often meets passionate opposition in the public. But rarely does the public debate fulfill the minimum standard of critically evaluating such complex issues. What is urgently needed are researchers who are independent from corporate support and who are willing to share their knowledge with society in an open dialogue. In this respect, Kevin Esvelt certainly is an outstanding example. While his view may arise controversial debate, it is, and this I'm sure of, motivating and stimulating to listen to him, to reflect on his ideas. Dear Kevin, we are very glad to have you here. Having enjoyed your TED lecture on YouTube, I am fully aware of the challenge it would have posed to you to speak from the pulpit of this historical alta aula, as it may be in contradiction to your passion for democratic spread of science. But as I have learned today from Tobias Just, you have already mastered the challenge, so now the stage is yours.
Thank you for the very kind introduction. And yes, I'm going to address all of you today from down here. I am here first because it is a great honor to be invited. But I'm also here because I believe that scientists should hold themselves morally responsible for all the consequences of our work. And as a scientist, I am deeply concerned that the power of our technology appears to be growing much faster than our wisdom. 5 years ago, no one would have imagined that we might be able to edit entire wild populations and associated ecosystems. Yet, based on laboratory results involving CRISPR gene drive systems over the past couple of years in several different species, it looks as though this will in fact be a possibility. I find this deeply alarming, although perhaps not for the same reason as many others. Many people feel that natural ecosystems are perfect as they are. And this is interesting because many scientists similarly believe that science is perfect as it is. And I am skeptical of both claims. Rather, I see CRISPR gene drive as a warning sign that the research ecosystem may not be up to the challenges ahead of us. The US National Academies released a report on gene drive last year in which they concluded that experts acting alone will not be able to identify or weigh the true risks and benefits of gene drives. And it's really unclear, if that's true of gene drive, why it would be any different for any other kind of powerful technology. That is, we commonly think of wisdom as typically arising from discussions among groups of people from widely varying backgrounds and different disciplines, much as the Marsilius colleague is intended to foster. And if that's true, then why does science typically take place behind closed doors, conducted by small groups of specialists in the same field, without much input from the outside world, and without disclosing, of course, what they're doing to the outside world until the time of publication when the technology is complete? This seems directly antithetical to an approach that would pursue wisdom deliberately. But at the same time, dare we contemplate changing how we do science? The scientific ecosystem is tremendously complex, just like a natural ecosystem. We cannot hope to anticipate all of the consequences of changing how we do science, and we rely on it, just like we rely on natural ecosystems. But at the same time, we don't understand everything about how the human body works, not even close, and yet we still practice medicine. That is to say, just because we can't predict all the consequences, that doesn't mean that we should not intervene. It just means that we should consider intervening when the benefits are great enough. But we should be cautious. So, as a biologist, anyone who works with biology knows that it is a complex system and things can go wrong. So I've learned a couple of rules that I try to abide by whenever engineering a complex system. Rule 1 is you always want to make the smallest possible change that you think can solve the problem. Rule 2 says, even so, you don't want to make that change everywhere at once. You want to try it out on a small scale, see what happens, and only if things work well do you then slowly scale up. Now, keeping this in mind, should we contemplate changing how we do science? Honestly, you have to quail a bit at even the thought. It seems like heresy, because the reality is that science and technology have been the main driver of civilizational progress and increases in human well-being over the past several centuries. But just because it's been successful doesn't mean the system could not be better. In fact, it would be very surprising if the research ecosystem was optimized for meeting our needs. And that's because no one designed it. It's an institution, and it's a cultural institution that has evolved from cultural practices and incentive structures that were laid down a long time ago without much conscious intent from any person. What's more, even though it evolved, it's an ecosystem. And we don't expect ecosystems to be adaptive in the Darwinian sense. That is, when you have a forest next to a prairie, you don't expect the forest to get better at being a forest because it has to compete with the prairie. That's not the level on which evolution operates. Similarly with the research ecosystem. So here you have this system that was never designed, and which we don't expect to have been adapted to past conditions, that now faces very different future challenges. Among other noteworthy differences, the cost of sharing information with other researchers used to be quite considerable. You had to put ink on paper, and then you had to ship that paper, a copy, to everyone who wanted to read it. Whereas, relatively recently, the cost of sharing information went to quite literally zero, assuming we could get the publishers out of the way. So, what we need from the research enterprise is technologies powerful enough to solve our problems, to keep our unsustainable civilization running, and hopefully put it on a more sustainable basis. We desperately need those. It's not like we can stop and stay where we are. There is no hope of that. But on the other hand, we need those technologies to avoid endangering civilization itself. Now, this is something that we haven't really faced recently, until recently, that is, but I think it's inarguable that if the equivalent of a nuclear weapon became widely available to a very large number of people, that would probably be the end. So there almost certainly are technologies out there. Maybe we're insanely lucky and there are not, but there probably are truly dangerous advances out there that we would best avoid. But it's hard to see how we can do that with the system as it currently stands. So the question then is, how can we reform the system? Because science is a culture. It is a powerful culture. It is an entrenched culture. And many of those who are involved in it are not particularly interested in seeing change. Many people have vested interest in the way it currently is, We need a way to change that cultural ecosystem. That probably means we need to somehow change the fitness landscape on which it operates. We need something large enough to shock people out of complacency and initiate changes. In other words, we need a technology for engineering ecosystems. And of course, with gene drive, we potentially have one. So I submit that gene drive may be not only the warning sign that we need to change things, but it may also be the key to changing the research ecosystem. Now, the story of gene drive really begins with dogs. So please, how many of you have a dog? Please raise your hand. Oh, come on, don't be shy. Audience participation, come on. None of you have a dog? Really? Really? Or at least used to have. Okay. Really? Okay, I'm shocked. Normally I get half the audience. All right, so when you had a dog, did you believe that your dog was the best dog? I mean, stand up for your best friend, right? Your dog could do anything, right? Did you ever believe that your dog could outcompete the wolves in the forest? Probably not so much. And that's symptomatic of a general rule. That is, we bred dogs for a very long time to do things useful to us, or at least aesthetically pleasing, or anyway, something different from what is necessary to efficiently reproduce in the wild. So we divert their resources. They're not as good at reproducing in the wild, and we expect natural selection, therefore, to weed out those engineered traits. And that means it doesn't really matter how you change the dog. That is, someone doing CRISPR genome editing on the dog, you're going to get the same result. It doesn't matter how you make the change. The point is, it's not something that natural selection is going to favor. Not an absolute rule. There are probably some cases where we could engineer a change that would be advantageous in the ancestral habitat, but it's going to be very rare. But with gene drive, of course, that potentially changes. So gene drive systems are natural. This is such a useful evolutionary trick that evolution discovered it hundreds of millions of years ago. And they are essentially ways of biasing inheritance in some way, such that a genetic element can spread through a population even if it doesn't improve the organism's ability to reproduce. Now, an absolute genius by the name of Austin Burt in 2003 proposed a credible way that we could harness one particular form of gene drive system, one that literally cuts its competing gene and copies itself in its place to engineer wild populations. And Austin was specifically interested in using this to combat malaria. Now, even if you feel that natural ecosystems are sacred as they are and should not ever be perturbed, you really have to pause and think hard when you consider malaria. Because in the time that I've been speaking, malaria has infected 5,000 people, and it's killed 10 children. So if we could do something to seriously strike a blow against malaria and perhaps one day eradicate it from the world, that's something that we have to seriously consider. It's worth noting that malaria is eminently natural. And for that matter, so is smallpox. We got rid of that, and I don't think anyone is sorry. So the problem was that harnessing these natural gene drive systems proves very difficult, just from an engineering perspective. So this is why I, in fact, had not heard of Austin's work when I first wondered what would happen if we tried to make CRISPR genome editing a heritable trait. That is, the ability to cut and copy it oneself as part of a CRISPR cassette. What would happen? Would that spread through a wild population, I wondered. So initially, when we developed CRISPR genome editing when I was in George Church's lab, we would deliver the CRISPR components on 3 separate pieces of DNA. One that encodes the nuclease, one that encodes the guide RNAs telling it where to cut, and one that encodes the sequence of the DNA that we want to insert in the genome. But what if instead you encoded all 3 of them on one piece of DNA, deliver this into the reproductive cells of an organism, CRISPR components will be produced, the nuclease will cut the target site of the genome very specifically, and they will all be inserted. And now, once one copy goes in, the cell will produce the CRISPR components on its own, meaning it's going to cut the other copy on the other chromosome and be copied over again. So now this organism will have 2 copies in its germline, which means that when it mates with a wild organism, all of the offspring are guaranteed to inherit 1 copy. And in those offspring, genome editing happens again. You cut the wild type copy and replace it. meaning that when these organisms mate, all of their offspring are guaranteed to inherit the edited version, as are their offspring, and the next generation, and the next, and the next. In other words, you've released a self-copying find and replace element into the population. So given enough generations, if it proceeds exactly this way, it's a little bit more complicated than this, then you will edit every copy in the wild, given enough gene flow. Now, this is in some ways tremendously promising. That is, we could use biology to solve ecological problems. Now we primarily use bulldozers. And when not bulldozers, lots of toxic chemicals. This is how we control pests. Indeed, most genetic engineering of crops has been to make them tolerant to toxic chemicals that we can then spray on the fields. This is potentially a way to tweak the pests, for example, so that they no longer like the taste and avoid the chemicals entirely. It's potentially a much more eco-friendly way to deal with ecological problems. But it's also something that only works in sexually reproducing organisms, exclusively sexually reproducing. Anything that can just divide asexually or self-fertilize, or even a lot of inbreeding can get around this. And it's something that is slow. It only spreads over generations, so it only works in fast reproducing organisms. And more worryingly, it is something that requires only transgenesis as the limiting step. Because CRISPR is now so widely available, there are, I hesitate to say this, but there are middle school labs that are using CRISPR to edit organisms. Not sexually reproducing ones, but it will come. So the limiting step is now, can you engineer a sexually reproducing organism? And anyone who can do that can potentially build a CRISPR gene drive. And that means that individuals can potentially affect the lives of many, many other people unilaterally. So what do you do if you see a technology coming that allows that sort of impact? Well, speaking from a bit of experience, you should tell someone trustworthy. Because no one person is wise enough to deal with this on their own. If this had been left to me, it would have been an epic disaster. But fortunately, I had many colleagues and advisors who were much wiser than I am, particularly George Church, who also introduced me to the ethicist he works with, Jean-Tine Lunshoff, and also Ken Oya, who's a political scientist at MIT, who specializes in synthetic biology and its implications. And Ken said, you really need to come to this workshop we're throwing on the ecological implications of synthetic biology, because this is probably the biggest one there is discovered to date. And at this workshop, we had ethicists, ecologists, government regulators, national security experts, and representatives of environmentalist NGOs. So we talked about it. All these people from different backgrounds and disciplines, and it was surprising how much we agreed on. Namely, three things. First, the security risks posed by gene drive are actually quite minor. That is, this isn't much of a weapon, and that's because it's very slow to spread, takes generations. It's easy to detect by sequencing. If you sequence the genome, you will see it. There is no way to hide it. And once you see it, you can build another one that will perfectly overwrite it whenever it encounters it. And it's not much of a weapon that is slow, easily detected, and trivially defended against. So we don't really have to worry about that too much. Second, the ecological effects. So what are the ecological risks of this technology for potentially altering ecosystems? It's going to vary on a case-by-case basis. Most changes to the genome would do nothing. Some would have major consequences, but it depends entirely on the species and what you do to it. So you can't make any sweeping generalizations. Life is tremendously diverse. Changes to life will have similarly diverse effects. And third, we needed to tell people before we ran any experiments in the lab. So that's what we did. We published 2 papers, one on the technical details of how it worked, what was necessary to make it evolutionarily stable, and perhaps most importantly, the necessary safeguards, given the likelihood of spread in the wild. And also an accompanying piece on regulation, essentially calling for broadly inclusive discussions, and for all research to be transparent, from the proposal stage onwards. And only then did we actually begin testing it in the laboratory, in this case in the safest organism, yeast, safest because they mainly reproduce asexually. And in yeast, we showed that it worked very well, and verified that all the safeguards we proposed worked. This was important, this notion of tell people before you run the experiments, because, again, the National Academies said it best, the best course of action is to ensure that those who would be affected by a proposed project or policy have an opportunity to have a voice in decisions about it. Not even saying a vote, but you need to give people a voice. And denying them that is really counter to our democratic aspirations. It's also probably deeply unwise. So given that, proceeding as we have been with research, by small groups of specialists behind closed doors, is arguably unethical. So that's what we've been doing. I think we handled that reasonably well, but we also made some pretty profound mistakes, particularly me. This is one of my greatest mistakes. This is figure 7 of our paper describing the technical details, and it's the one where we envisioned all the sorts of things you could do, solving ecological problems with biology. And at this time, I was thinking pretty much entirely about this is a way to make a very small change to one species to solve a major problem without affecting any others. Smallest possible change. That's rule 1. But I hadn't really thought enough about rule 2. Because rule 2 says start small and only scale up if warranted. And this is a technology that scales itself. And that means that this very pretty Venn diagram that I drew, yeah, not so much. Most of those applications are just not even remotely feasible. And this is hard because now many people, especially scientists, have sort of internalized that all of those things are possible with this form of gene drive system. And now I don't believe that they are. So how invasive are these systems really? Like is this so much of a problem? And we've done some modeling of this. We took the worst drive system published to date. It's in fruit flies. It only copies itself approximately half of the time. It's not designed to be evolutionarily stable. And that means it only goes to about 30% of the population before the frequency starts going down. But the number of organisms you have to release into a population to get it to go up to 30%, well, if you release 2, then there's about a 50-50 chance that it will manage to take off and will not stochastically go extinct. If you release a dozen, it's a near certainty. And of course, if you go up to 30% of a population and there's pretty much any degree of gene flow with a neighboring population, you're going to get at least a few organisms moving over, and it's going to go to 30% there, then it's going to go to the next one, and the next one, and the next one. In other words, this is extremely invasive. You should think of it as like introducing a rat to an ecosystem that did not previously have rats. It is on that level of invasive. It will only spread through the target species. There has to be gene flow. But again, even if there isn't natural gene flow, what are the odds that some human, somewhere, is going to decide to move it deliberately? How many trolls are there on the internet? So we have to assume that it's going to invade all populations of the target species. And that means that it's really hard to run a safe field trial. And if you don't run a safe field trial and you have to assume that it's going to spread across every international border that's relevant, it's going to be really, really hard to get people to agree to this. This is not necessarily a barrier to using gene drive against malaria. And this is incredibly important because Austin is pursuing that and has an excellent chance of success, I think. But that's only because malaria is such a tremendous, horrific problem that you could sum every possible negative side effect that has been dreamed up in all of the risk assessment workshops to date by experts and members of the public, and including many African scientists and African community leaders, and put them all together, assume that they all happen, and the harm done by all of those together wouldn't come close to that done by malaria. I have 2 young children. If they were at risk of malaria in Africa, I would vote, go ahead, never mind with the field trials. This is so bad that we need to do it immediately. Now, I don't get a vote because I don't live there. I'm not sure that anyone who doesn't live there should get a vote, but I would hope that the African nations who would be affected can have a good discussion and potentially come to agreement. The danger, of course, is that someone is going to say, well, you don't have the right to prevent me from protecting my own children. But the hard thing is if one country decides to go ahead and release it before the others have agreed, this doesn't spread that fast on its own. If you want to eradicate malaria, you would need to release these evenly distributed over the whole population. And so if you do it without the permission of some countries, they're not going to let you do that in all likelihood, even if it works. And that means you could net lose lives relative to if you waited. So it's hard. It really does seem to be a consensus among everyone who would be affected. Only possible because malaria is that bad. But that means that except for malaria and maybe a couple of other problems like that, this system is just too invasive to be useful. And this is a really hard realization to come to. Because this means that we have to worry about the downside. And there isn't that much of an upside of this technology as it is now. What was the downside? Well, here's the nightmare scenario, and this is something that I communicated with Austin and we corresponded with extensively. Suppose you have all of these labs that are now using CRISPR for the first time. Suppose you have a fruit fly lab, and they think, wouldn't it be nice if we could knock out both copies of a gene in a single step reliably? Wouldn't it be nice if we could maintain that knockout very easily in our laboratory populations? Hey, we could insert the CRISPR nuclease and guides and do that. Wouldn't that be genius? And it would be. But they might not even think of the fact that this could spread in the wild. So Austin and I corresponded about this, but we just couldn't think of a way of preventing it. Because of course, if some lab was planning on doing this, they're not going to tell us or anybody else. Because after all, if they did, then that would risk somebody else taking their idea and throwing more hands and money at the problem, publishing first and claiming all the credit. So of course they were not going to do that. And that is why it happened. But we were very lucky, because first of all, the grad student involved in the project did think about it and use some extra precautions. They targeted a gene that's pretty costly in the wild, and because of some combination of those factors, there was no accidental release. On the downside, when they wrote it up and sent it to the journal, it got through 2 senior editors familiar with our work. The scientists doing it had not seen it, because not everyone reads every article in Science and eLife or even the New York Times. But it got through 2 senior editors and a reviewer and an editor that were aware of our work, and this is the very same journal in which we had published our call for safeguards. So that's a little bit worrisome. But George was asked to comment on a copy by a journalist, which allowed us to contact the authors and explain our concerns, and they agreed to edit the discussion to emphasize the need for safeguards. And later on, they joined us and a bunch of other scientists, including Philip, who's in this room, to agree on some consensus recommendations for which safeguards should be used by labs interested in pursuing this kind of research. It's not binding, but at least it's out there. And it's been echoed by the US National Academy and various other advisory boards. So, in some sense, that's inspiring. On the other hand, it's a little worrisome, right? This is how things can go wrong with technology. But what are we really worried about? Would this have any effect if it did get out? And the answer is almost certainly no. There would be no ecological impact of that drive system or any of the others that have been made in the lab to date if they were to get out. So what's the worry? Well, the risk isn't ecological, it's social. Because I believe in this case, a single laboratory accident involving a gene drive system would be very well publicized. The headline would be on the order of, scientists accidentally convert entire species to GMOs. Is CRISPR to blame? And that would be very bad for public trust in science. It would fairly conclusively prove that scientists cannot, in fact, be trusted with technologies this powerful. It might lead to restrictions on science that would block advances that civilization actually needs. And it would tremendously damage society's trust in institutions, which is already perilously low. And that's apart from the direct impact. A single death in a clinical trial set the field of gene therapy back by a decade. Similarly, if you imagine the cost in delaying the potential application of gene drive against malaria by a decade, that would be deaths, mostly of children under the age of 5, in the millions that could otherwise have been prevented. I do not want that on my conscience. So what can be done about it? This is a system that is out there, it is possible, it is the easiest form of building this, and it keeps spreading indefinitely. But that's the problem. Is there a technical solution that would allow us to realize a lot of those benefits and potentially solve some of the risks? In other words, is there a technical way that we can make these drive systems local, rather than having to get every country that harbors the target species to agree? Can we just confine it to a particular community so that one town can decide, do they want to alter their shared environment together or not? How do you do that? The problem with a CRISPR gene drive as it is it has everything it needs to copy itself. So the solution is to separate those elements into multiple pieces, scatter them on the different chromosomes. So we call this a daisy chain drive because the elements are linked. Element C causes element B to be copied. Element B causes A to be copied. But C is like a normal engineered gene. That is, it's never going to increase in a population. You release these organisms out there, and C is never going to go up or down. It's just genetic drift. But while it's around, B is going to go up. While B is around, A is going to go up even more. So you get amplification. And when you add selection, because whatever change we make is going to be opposed by natural selection, it's going to drag it all down. So the net effect is kind of like a multi-stage genetic booster rocket. That is, the different stages propel the cargo higher and higher through the population, but eventually it will come down because it's only local and it's costly. It will run out of fuel. What's more, you can add more links to the daisy chain, and that's like adding more fuel, and so it becomes progressively more powerful. How much more powerful are we talking about? Well, if you model it, if you assume on the low end of copying efficiency in the organisms described to date, about 90% copying efficiency, which is definitely on the low end for the mosquitoes in particular, you can release one daisy drive organism, and in 20 generations it will have gone to fixation in the local population. If you have 98% homing, which is more common amongst the malarial mosquitoes, then it's 1 per 500. So this is potentially very cost effective, but it's still ultimately limited. It does run out of fuel and it stops. So this is a daisy chain form. There's another way to do it as well, which is you simply have two stages, B and A, but you add lots of copies of B around the genome. Every time it mates with wild type, half of them go away. So eventually it runs out. So we call this a daisy field. And we're working on both. But neither of them solves this key problem of gene flow across political boundaries. Because the harsh reality is, town A decides that they want to alter their environment. Their next door neighbors in town B don't want to. How do you convince the engineered organisms that they're not supposed to cross that invisible boundary? This sounds impossible, but what you really need is for the genes to hold a vote. That is, you want genes in the majority. If they're in the majority, that probably means they're supposed to be there, and you want them to go to 100%. But if they're in the minority, they're probably not supposed to be there, and you want them to go to 0. You want natural selection to wipe them out. And it turns out that there is a genetic phenomenon where exactly this happens. So if you make it bad to be a hybrid, it's bad to have one copy of the engineered gene and one copy of the wild type, then you want to be in the majority. Because if you're in the majority, you're more likely to find a mate just like yourself and not make a hybrid. Whereas if you're in the minority, you're very likely to make a hybrid. So it has exactly this effect. And so we figured out a way to link this to a daisy drive, so we call it a daisy quorum. And the idea is it spreads in the target population, runs out of daisy elements, and then if it's in the majority, it takes over. If it's in the minority, say because it's gone into town B, the natural selection will wipe it out. So that will ensure that town B stays largely wild type and town A stays largely engineered. What's even more exciting is this is a potential way of removing every last copy of an engineered gene, because the further you go into the minority, the harder and harder it is to find a mate who won't make a hybrid, and so the stronger the selection against you. And this is what we need in order to remove all copies of an engineered gene from the population. That is, if you use a daisy quorum combined with a suppression drive system, you can accomplish what we call a daisy restoration, which would allow you to remove any engineered gene, even a standard CRISPR gene drive, potentially, from any drive-amenable population, and restore it to wild type. That is to say, even if somebody screws up, it's more like graffiti. You just wash it off. Theoretically, this will work in any drive-amenable population, which is really exciting because it means it's not just cleaning up after gene drive systems, it's also any other engineered genes that move into species they're not supposed to be in or move across borders and so on. We could potentially restore all those populations to wild type if that's what people want. And since many people do not agree with me about the intrinsic value of untrammeled nature, I have to respect those opinions. And so we should let people decide. If that's what they want, then we need to, as scientists, need to give them that capability if at all possible. So overall, I think, or at least I hope, that this will correct my earlier error, and that version 2 of the Venn diagram will be true for local drive systems, such as daisy drives. Maybe there are better ways of doing it. I hope someone comes up with something even better, but maybe this will be enough. And if you want to learn more about it, we're building a site called Responsive Science. Our lab webpage has a lot of information if you want to see the more technical details, and you can look up our preprints. But Responsive Science is where we hope to encourage an actual discussion and invite concerns and criticism from everyone that could actually shape our experiments. And our goal for this is really a new form of doing science. That is, we want researchers, especially those working on technologies with shared impacts, to openly share their proposals. That is, you're already writing the grant. Scientists already do that. You send it to the funder. Instead of just doing that, make it publicly available. That's what we want. Actively invite concerns. Don't just stay behind closed doors and hope that people don't find out and then the activists won't show up. Actively invite concerns and criticism and respond to them. Request community guidance of a local community, ideally one that would be affected by the technology, because they might think of something that you won't. And arrange for independent assessment. The last person you want to test a hypothesis is the person who proposed it. The same for testing a technology that someone has invented. And finally, start local and only scale up if things look good. Safe, controlled field trials. How do you do this, though? This is really new. There have been a couple of projects that have been trying to do this. Eliminate dengue and Austin's Target Malaria have been working on this, particularly in Africa. But we wanted to, frankly, tackle an easier problem. So we're working close to Boston with island communities off the coast of Nantucket and Martha's Vineyard. And this is comparatively easy because these are well-off communities with incredibly well-educated populations. And the goal is to prevent tick-borne disease because almost half of people who live there have been impacted by tick-borne disease. Particularly Lyme disease, but also others. But it's a tremendous, tremendous problem, and there aren't any good solutions. What's unique about Lyme disease in the eastern United States, which is unfortunately not true here in Germany, is that almost all of the ticks are infected by when they bite one particular animal, the white-footed mouse. The white-footed mouse is just by far the best reservoir of all the different pathogens that these ticks carry. So our idea is if we can heritably immunize all of the mice in the population, then we can prevent almost all of the ticks from becoming infected and then prevent almost all cases of disease. And it would be a stable and lasting change. So this is the idea. We called together a workshop in which we had ecologists, ethicists, regulators, and local community leaders from these islands and said, how should we go about doing this? And then implemented those recommendations by going to town hall meetings on the islands. And somewhat surprisingly, for a proposal that would involve releasing up to 100,000 genetically engineered mice onto each island, the people were quite enthusiastic. In large part because it would be open, nonprofit, and they would have absolute control. That is, if at any time they say no, we walk away. That's it. And frankly, I won't be sorry at all if at least one of them says no, because I think that's an example that needs to be made, that the scientists really will walk away when the community says no. But right now, both islands have nominated steering committees that will actually be in charge of the project. We are just the technical hands. And the citizens have made it very clear that they want the mice to be resistant to not just Lyme disease, but also to tick bites themselves, which will block most of the other diseases. They want both. They say if you're going to engineer it at all, do everything. But they don't want any foreign DNA in those mice. So I always explain the concept of a species is totally artificial. It's a human box because our minds like to put things in categories. But even biologists can't agree on what a species actually is. There's gene flow between what we call different species. Technically, lions and tigers are different species, and yet if you put them in a zoo in the same cage together, a male and a female, well, guess what happens? So you get tigons and ligers. So, okay, but that doesn't matter. That's the scientific reality that species is arbitrary. The cow genome is 25% snake due to a gene transfer event a couple of million years ago. All sweet potatoes are GMOs, dating to the dawn of sweet potato domestication. It's all immaterial because the people said that they don't want foreign genes in their mice, so there's not going to be foreign genes in their mice. And that also means no gene drive. And because they're islands, there doesn't need to be. We can just release enough engineered mice using 100% mouse DNA, and that will be enough. So we hope that this will set an example. We hope that our daisy drive systems will work well in the laboratory and the worms where we're testing them. And we hope that this will solve the various problems that this particular box has unleashed. But we're still missing the larger picture, the larger problem here. And that is that the research enterprise encourages closed-door experiments that don't invite scrutiny from those outside the particular discipline. That is to say, the incentives are in many ways antithetical to wisdom. So how do we change that? Well, how is gene drive the key? We have to defy these incentives somehow. So my group is doing this. We pre-register all of our gene drive experiments. But right now, we're the only ones. And we can do it because we have privileges, namely I'm at the media lab, where the incentives are somewhat different, that the other labs don't have. They really do face that risk of being scooped, in particular their students, and it's real. I know just about everyone in the field, and the consensus is, look, we'd be happy to be open about it. We agree with you that there would be tremendous moral and practical benefits to doing that, but we have responsibilities to our students. We can't do it unless everyone has to do it. So the only way forward is to change the incentives for the entire field at once. So how do you do that? Journals, step one. If the journals require academics to pre-register their experiments in order to eventually get published, if Cell and Nature and Science all require this, plus all the secondary journals and so on, next tier down, you can bet the researchers will do it, particularly the academics. Funders. If the funders said, look, your grant proposal is going to be public as soon as you send it in, or at least as soon as we fund you, and that's a condition of funding, well, yeah, we kind of have to say yes. Without the funding, the research doesn't get done. If the regulators say, you have to be open about this and pre-register what you're going to do, then we kind of have to do it. That's the law. But all of those face the problem of the collective action problem. There's lots of journals, there's lots of funders, there's lots of countries. The incentives only change if most all of them change. And none of them want to do it unless everyone wants to do it. In other words, we just move the entire problem back one level, from the researchers to the incentivizers. But there is one last lever that we might pull that doesn't require all of that. And so we're exploring the possibility of using intellectual property, which sounds very counterintuitive, right? Especially for someone who is ostensibly as anti-corporate as I am. But intellectual property was originally designed to ensure that people shared the details of their invention so that others could build upon them. In practice, you never disclose enough in a patent to allow anyone to actually duplicate it. But that's the intent. And here's a what-if scenario. What if a nonprofit entity was created and given patent rights to enough key intellectual property in a field such that, if required, countries where there is no strong research exemption, that is, where you technically need a license to use the technology, even if you're a nonprofit academic, and the United States is very much such a country, as are many others, then the nonprofit could say, we'll give you a license for free, but only if you pre-register and use appropriate safeguards to ensure that your gene drive doesn't get out of control. Because after all, if it gets out of control, then our IP is probably worthless, or at least severely damaged. And there's the incentive that makes it hold up in the courts. You have to work with the system that you have. And so this is sort of our last Hail Mary to try to get the ball rolling. Because if the IP can move the system most of the way there, then it's much easier for the journals and funders and potentially the regulators to come on board. And then if we can do this for gene drive, then that's how we're abiding by Rule 2. We'd be trying out open and responsive science as a field trial in the field of gene drive, and we can see how it works. Because things could go wrong. There will be winners, there will be losers. We have to figure out how to compensate the losers somehow. Rough edges. The system will have to be tweaked, and we have to figure out what to do, and we should do that in one field. And if it works in one field, then perhaps that can spread, not so much like a gene drive, but to other fields where there's shared impacts, and potentially to applied science in general. This is not crazy. All clinical trials already require pre-registration. There's a movement in psychology to move towards pre-registering experiments just for reliability reasons. It's in applied science, where you have intellectual property, potential financial returns, and startups, that is the biggest barrier. But if we can do it here, and gene drive is uniquely suitable because, particularly with a standard gene drive system, there's not much commercial incentive to build something that will solve the problem everywhere in the world. It's not exactly a good return on investment. So here's where we have a unique confluence of moral and practical reasons to do something differently. And if we do it, then we'll have abided by our self-imposed rules for engineering complex systems. And more broadly, I'd like to conclude by just saying, if we're going to even contemplate engineering natural ecosystems, then we should begin by cultivating wisdom in science itself. Sincerest thanks to all of my colleagues over the years who have provided me with advice far wiser than I could generate myself. To my students, who are the real heroes because they are the ones putting their ideas out there. And to our funders for allowing us to do the work, in particular to the organizers and for the very kind invitation, and to all of you for your attention.